So now let me discuss the various methods of the gene transfer. So first is transduction. So if you take this transduction, here it is the transfer of resistant gene through the bacteriophage, right? So if you see this transduction, it is the transfer of resistance gene transfer of resistance gene through the bacteriophage. Right? This is what is the transduction. Now, let me tell you the antibiotics which will undergo the transduction. The antibiotics which will undergo the transduction is, example, penicillin will have the development of resistance within the bacteria by the transfer of resistance gene via the bacteriophage. Next, apart from penicillin, you see the other antibiotics. The other antibiotics like erythromycin, will develop resistance via transfer of gene via the transduction, and the other antibiotic is the chloramphenicol. The other antibiotic is the chloramphenicol. So remember, transduction, it is the transfer of resistance gene through bacteriophage. The antibiotics are penicillin, erythromycin, and as well as the chloramphenicol. Now let me talk about the conjugation. Let me talk about the conjugation. Now, if you see this conjugation, it is due to the physical contact between the bacteria that is responsible for multi-drug resistance. So this is due to physical contact between the bacteria. It is due to physical contact between the bacteria that is responsible for multi-drug resistance. Now, among the various mechanisms, this conjugation is very important mechanism for the development of resistance against chloramphenicol, against chloramphenicol and the other antibiotic, that is streptomycin. Other antibiotic, that is streptomycin. So conjugation, it is due to the physical contact between the bacteria and that is responsible for the multi-drug resistance. And this is a very important mechanism for the development of resistance against the chloramphenicol and streptomycin. Alright, so this is about the conjugation. Next, we have another method of transfer of the gene resistance which is called as the transformation. Which is called as the transformation. Now, so if you see this particular transformation, it is the transfer of resistance gene through the environment. So this is the transfer of resistance gene through the environment. Transfer of resistance gene through the environment is called as transformation. And this particular transfer is not significant clinically. right? Not significant clinically. The example is penicillin G, right? Example is penicillin G, right? So these are the various methods of the gene transfer for the development of resistance. One is transduction, conjugation and transformation.